Today, we're diving deep into sickle cell disease. We're going to unpack how a single tiny error in the genetic code, think of it as molecular sabotage, can unleash a lifetime of consequences.
I mean, think about that for a second. That's not just a dramatic question. It's the fundamental reality for millions of people around the world. It's the story of how one tiny mistake at the molecular level can completely undermine the very cells designed to carry oxygen and life. So, yeah, let's get into it. All right, here's our game plan. We're going to start with that single genetic letter, then follow the domino effect, the molecular cascade of chaos it causes. From there, we'll look at what a crisis actually looks like inside the body, understand the long-term toll it takes, and finally, explore the incredible new science that's fighting back. To really, truly understand sickle cell disease, you have to go right back to the beginning, back to the source code, our DNA, where this whole story begins. Okay, so what is it exactly? At its heart, sickle cell disease is a genetic blood disorder. Now, it's passed down in a pattern called autosomal recessive. All that means is that to get the disease, a child has to inherit the faulty gene from both parents, not just one. And the error itself, it's a tiny typo in the beta-globin gene, which is basically the blueprint for a crucial part of hemoglobin, the protein that does all the heavy lifting of carrying oxygen in our red blood cells. So the big question is, how does one microscopic genetic typo cause such widespread chaos? Well, it's a four-step molecular chain reaction, a cascade of sabotage that starts with that single letter. Okay, let's walk through this. Step 1, the typo. On chromosome 11, the DNA sequence GAG gets misread as GTG. That one little switch leads to step 2, a faulty protein. A water-loving amino acid gets swapped out for a water-fearing one. And this is where the trouble really starts. This leads to step 3, polymerization. When this mutated hemoglobin, we call it hemoglobin S, drops off its oxygen, that new water-fearing spot makes the molecules incredibly sticky. They start clumping together, forming these long, rigid rods. And that brings us to the final step, number 4, the sickling. These rods literally warp the red blood cell from the inside out, twisting its nice, flexible disc shape into a hard crescent or sickle shape. And this is the absolute key to the whole disease. You swap out one molecule that loves water for one that hates it, and you've created a sticky patch on the hemoglobin molecule. So anytime oxygen levels drop, these sticky patches desperately try to hide from the water around them by glomming onto each other. That's the moment the whole destructive process kicks off. All right, so now we have these misshapen, rigid cells floating around, and they immediately start causing two massive, simultaneous problems all throughout the circulatory system. This image really lays out the two core problems perfectly. Let's call them the twin pillars of sickle cell pathology. First, you've got vaso-occlusion. That's just a fancy term for vessel blockage. See how these stiff, sticky cells are creating a traffic jam? That's it. They literally form log jams in small blood vessels. The second pillar is hemolysis. As you can see with that ruptured cell, these sickle cells are incredibly fragile. They break apart way, way faster than healthy cells, and this leads to a chronic shortage of red blood cells. In other words, anemia. And once you understand those two mechanisms, the classic early symptoms of the disease make perfect sense. Jaundice, that yellowing of the skin and eyes, that happens because all that hemolysis, all that cell destruction, releases a yellow pigment called bilirubin. The constant fatigue, that's a direct result of the anemia from hemolysis. And dactylitis, that painful swelling in the hands and feet you often see in infants, that's a textbook example of vaso-occlusion, where tiny blood vessels in their small bones get blocked. Now, the most famous and feared part of this disease is the vaso-occlusive crisis, or what we call a VOC. This is when that vessel blockage gets so bad that it cuts off oxygen to tissues. This is called ischemia, and it results in episodes of absolutely excruciating pain. These crises are, without a doubt, the #1 reason people with sickle cell end up in the hospital. And these painful crises don't just happen out of the blue. They're often set off by common triggers, things you and I might not think twice about, like a simple infection, getting exposed to cold weather, not drinking enough water, or even just going through a period of high stress can be enough to ramp up the sickling process and kick off a full-blown VOC. Over time, this constant, relentless cycle of blockage and destruction takes a devastating toll. It leads to the slow, cumulative damage across pretty much every organ system in the body. And this timeline, it really drives home just how progressive this disease is. You can see it starts early. In infancy and childhood, you get things like splenic sequestration. But then as patients get older, moving into their teens and adulthood, the really severe complications start to show up. Things like stroke, chronic pain, and pulmonary hypertension. It's just a lifelong, relentless assault on the body.
So let's connect these complications back to our twin pillars. A stroke? That's vaso-occlusion in the brain. Acute chest syndrome? A life-threatening lung issue? Vaso-occlusion in the lungs. Chronic kidney disease? Same story, blockages in the tiny vessels of the kidneys. Pulmonary hypertension is a bit different. It's actually driven more by hemolysis. All those destroyed cells release hemoglobin into the blood, which sucks up a molecule called nitric oxide. And since nitric oxide is what keeps blood vessels relaxed, losing it makes them constrict, causing dangerously high blood pressure in the lungs.
For decades, all we could really do was manage the symptoms. But now we have this incredible, expanding arsenal of therapies that go after the disease's core mechanisms. We're finally fighting back.
Let's look at the main players. For a long time, our best weapon was hydroxyurea. It basically tricks the body into making more fetal hemoglobin, which gets in the way and stops the sickle hemoglobin from sticking together. Then there's voxelotor. It also stops the sticking, but in a different way. It makes hemoglobin hang on to oxygen tighter, keeping it in the non-sickly, relaxed state. And then you have crizanlizumab, which attacks the problem from another angle. It targets vaso-occlusion by blocking a protein that makes the cell sticky, preventing the logjam before it even starts. But honestly, the most exciting developments are happening right now. For the very first time, we have therapies that don't just put a Band-Aid on the symptoms, they aim to go in and fix the root genetic problem itself. Just recently, 2 of these groundbreaking gene therapies got approved. You can think of them in 2 different ways. LifeGenia is like adding a new, correct instruction manual into the cell. It uses a modified virus to deliver a healthy copy of that beta-globin gene. Casgevy, on the other hand, is like a biological search and replace tool. It uses the revolutionary CRISPR technology to go right into the patient's own DNA, find the problematic section, and edit the code directly. It's a total paradigm shift from lifelong management to a potential one-time fix. And as Jennifer Doudna, who co-invented CRISPR and won a Nobel Prize for it, says, it's about changing the very code of life in a precise way. I mean, for a disease caused by a single wrong letter, this kind of precision is the ultimate goal. It's just incredible. So, if you're going to remember anything, remember these 5 points. 1. It's a genetic disorder caused by a specific mutation. 2. The core problem is that hemoglobin clumps together when it loses oxygen. 3. This leads to our 2 pillars, vessel blockage and cell destruction. 4. A key drug, hydroxyurea, works by boosting a protective form of hemoglobin. And 5, brand new gene therapies are now offering the potential for an actual cure. And hey, here's a little clinical trick to take with you. A super easy way to remember the common triggers for a crisis is the mnemonic SICKLE. S for stress, I for infection, C for cold, K for kidneys, which reminds you of dehydration, L for low oxygen, and E for exertion. Simple, right? And that really brings us to our final thought. The journey of understanding sickle cell, from a single letter change in DNA to a therapy that can precisely edit that very code, it really mirrors the journey of modern medicine itself. So the question we're left with isn't just if we can cure these diseases anymore, it's how this incredible new power is going to completely reshape our future.